Welcome to our 5th virtual session, I believe. We are going to be doing a liver enzyme lab today, which is going to, I gotta mute somebody here and make myself the pinned video. Okay, we're going to be actually using chicken livers to model how enzymes work inside the body. Right, and you've read about enzymes very vaguely, right? There's not a ton of information in, I'm just gonna mute you guys and so that we don't have all this background noise here. So these enzymes are a really important part of the body and how the body works. So we're gonna learn about how they work today, doing a lab, because that's of course the best way to learn about how stuff works. So we got some chicken livers here, which people eat, right? We're not going to eat them, but we are going to talk about them. So I'm going to go ahead and share my screen. I did make a PowerPoint today. Jordan, I don't know if you want to come sit on the other side of me so you can see it. It's not very much information on it, but so you could be following along here. So our liver enzyme lab, our objectives are going to be to model enzymes working in a cell by using an organ filled with enzymes, i.e. the liver. Your liver is also filled with enzymes, right? Describe the relationship between enzymes and temperature. And we're also going to prove that the reaction is happening, right? So you may notice a theme this year. There's a lot of proving going on, right? Because that involves higher order thinking here, like having to think about how we could actually prove that something is happening. So our warm-up today is going to be to look through the background of the lab, right? Here is the lab. I'm going to go ahead and post the link, into the chat, just in case you have not looked at it yet. Some of you have already done the background. That's awesome. Just look through it again, right? Specifically looking at these 3 words, because those are the 3 words that we're going to focus on the most here. And I would definitely make sure that you are understanding what reaction we're going to be doing. I'm going to go over it, but regardless, let's spend about 3 minutes looking through the background of the lab. It's not too much reading, all right? And I'm going to be silent. In fact, I'm going to get ice while we are doing that. I guess I'll take my computer with me. You want to come get ice with me, Jordan?
Yeah, sure.
All right. Jordan's going to help me get some ice while you guys are reading. Nice cold ice.
Yep.
All right, so if you need a little bit more time to read, please type a 1 in the chat. If you're good, you get the gist, you're ready to move on, type 2 into the chat. Okay, we got a mixture going on, so we'll give another minute here. And Jordan and I are going to be working on setting up the lab. Okay. Would you like to cut up the liver? Yes. There's gloves. You don't have to actually touch it unless you want to. Here's some gloves. So we're going to need 5 pieces that are as close to the same size as you can get them. Right? It doesn't have to be exact, but relatively the same size. And they need to be able to fit in these test tubes. Okay? So pretty small pieces.
Small pieces.
Okay, so Jordan is going to start getting set up here just because we are going to have to heat water up. And that does take, you know, some minutes to do. So basically what we're going to be doing is taking equal size pieces of this liver. You guys can take a little glance at how it looks, right? It looks like a liver. That's actually how your liver looks, although your liver is way, way bigger than that. So we're going to be looking at this reaction that happens. So basically, your cells are always producing hydrogen peroxide, right? Which you guys might know as the painful thing that your parents probably poured on your cuts, right? It's a poison to your cells. So when it's produced, the cells need to break it down as quickly as possible, right? Otherwise, it's going to kill the cell. So an enzyme in your body called catalase, it's also in chickens and other animals, right, breaks down the hydrogen peroxide into water and oxygen, which is great. We like oxygen, right? And water, also harmless. So what we should see, right, if the reaction is happening is what? What kind of stuff do we see when there's a reaction? That's perfect.
Okay.
Bubbles. Excellent. Thank you, Ariella. Bubbles, right? We should see when we pour the hydrogen peroxide on the liver, we should see bubbling, right? And the amount of bubbling that we see is actually going to be telling us really how much reaction is happening. If we see a ton of bubbling, that means it's like going crazy. If we see a little bit of bubbles, a little bit is working, right? So we have to think here, there's hundreds of thousands of enzymes inside these tiny pieces of liver that we're going to use, right? There's not just one, there's a bunch. So some of them might be working and some of them might not be, and we'll kind of talk about why. So the most important thing really to understand about an enzyme is that their shape matters, right? So think about a Lego. You're building a castle, you have your last piece, and your sibling melted it a little bit with a lighter, and now it doesn't fit, right? Sounds like a real possible story there. Very frustrating, right? You can't fit the Lego in. It can't do its function. The same thing with the enzyme. If the enzyme's shape changes even the slightest bit, they don't work, right? They actually will just completely stop working, and they can never come back from that, right? It's like they're ruined, and then that's it. So that's part of the main reason why your body works so hard to keep certain conditions, certain temperature, certain salt, certain pH, right? Those things are not fluctuating all over the place. And one of the main reasons is enzymes, right? Not just catalase, but every enzyme in your body requires specific conditions. So when an enzyme is ruined, right, its shape is changed, it's called denatured, right? That's just the word that's associated with it. You can think of it like its nature is undone or ruined. All right, so if you haven't done the background questions yet, we're going to talk about them right now. Okay, so the first one is, which gas will be released if the enzyme is working, and how could we measure it? So looking back at the equation, what gas is going to be produced here? Is Ariella the only person alive today? Oxygen, thank you. Okay, there we go, there we go. So oxygen is the gas that's gonna be produced, right? Do you guys have any idea? How could we measure it? Can we measure it? We're gonna talk about how we could measure it in a little bit, right? There is a way we could prove oxygen's there. If we had fancy technology, we could use a probe. There's actually probes that detect oxygen levels, right? So we can hold it over the reaction and see that the oxygen level is going up. Okay, so we're gonna be looking for that gas that's produced, which is gonna make those bubbles that Ariella told us are gonna happen. We're also gonna be using 4 different temperatures for our liver, right? We're gonna have ice cold in our ice. We're gonna have room temperature. We're gonna have body temperature, which is gonna be as close to 98 degrees as we can get it. And then we're also going to have boiling, right, or 100 degrees Celsius. So, body temperature, room temperature. So part of this is gonna be you guys predicting which one of these situations you think is actually going to be the best for the reaction. How are we going to get the most bubbles? I guess the hot doesn't work on this one. Try to let this water warm up a little bit so it's not so chilly. Okay, there's like slime left over on that sink. So we are gonna have our 4 different temperatures with our liver, and what do you think we could use as a control? What were the temperatures again? I'll repeat them, give me 1 second. It's hard for me to talk and spill stuff out with water at the same time. All right, our 4 temperatures are ice cold, right, just ice water. Ideally, it's gonna be 32 degrees, right, close to freezing. Room temperature, which is just gonna be the temperature that the water came out. Hopefully it warms up a tiny bit. Body temperature, right, we're gonna warm it up to, it's 37 degrees Celsius, 98 degrees Fahrenheit, and boiling, right, so actually getting the water to boil. So I'm gonna start heating these waters because otherwise we will never finish this lab. And Jordan and I are gonna be monitoring the temperatures here as we go through this. But what we need to do first, before we can even start measuring or putting the livers in here, is we need to figure out which one is going to react the best. And what we could use as a control. So what do you guys think? How could we do something to test this? Oh, good thought here by Alexandria. She says, could we use the same thermometer each time we measure the water and use that as our control? Yeah, so that's gonna be part of it, right, is using the same materials. The liver, we're gonna try to have it the same size, right? We can use the same thermometer. I have 2 of them here. And we'll pick whichever one is the best. Really, our control is just going to be that we're gonna pour hydrogen peroxide in an empty container, right? Or into water even. Because really, we wanna show that the hydrogen peroxide is not bubbling by itself, right? Because imagine if the hydrogen peroxide just bubbled no matter what. Well, then this experiment would be a waste of our time, right? Because it would just bubble no matter what liver, what temperature, what we did. So we want to avoid that. So we're going to just pour hydrogen peroxide into an empty test tube to show that it doesn't bubble. And that's going to be our control. Okay, so just plain hydrogen peroxide. We could also pour it into water if we want, right? But really we want to show those bubbles are not going to happen unless the liver is present. So these words you can define later. All right, why are enzymes important? You guys are gonna answer that yourselves. What does it mean for an enzyme to be denatured? You're gonna answer that yourselves. And then what environmental factors affect enzymes' function? I will help you out with this question number 6 here, right? pH is a huge one. Where am I typing? There we go. pH is one, and also temperature, which is something that we're going to be looking at in this lab. Another one is salt, right? And there are other factors like the amount of substrate or what the enzyme actually breaks down, because if there's no hydrogen peroxide, then no enzymes are going to be working. Okay. So next up is the prediction phase. Which temperature do you think will produce the most oxygen and why? Right, and there are the temperatures listed out there on the lab: room temperature, ice cold, boiling, body temperature. I put them in Fahrenheit, right? We will actually be measuring them in Celsius because we have Celsius thermometers. Right now, it looks like our ice cold water is around 3 degrees Celsius, which we would like it to get a little warmer, but that's not the worst. And oh, this one's kind of dirty. Shouldn't affect it actually, but. All right, these are the pieces right here? Yeah. Okay. So we're gonna take our liver, which by the way, liver feels very squishy. And we're gonna be putting it in these test tubes, right? We don't wanna put the liver directly in the water because we just want the liver to heat up. We don't want it to be compromised by anything else. And we don't know if, for example, water might affect it, we don't know, right? So we wouldn't wanna just dump it in our cup of water.
Since the liver is naturally inside of a warm body, would it, like, the control would be it's on the body temperature, because that's how it reacts naturally?
That's a great question. So Jordan just said, wouldn't the body temperature kind of serve as a control? Possibly, right? We're coming into this lab assuming that we don't know if that's a normal situation for an enzyme, right? Because for all we know, maybe it works well in body temperature, but maybe it works better in other conditions, right? But what you're saying is a great thing you could say for your prediction personally, right? That perhaps you think body temperature will be the best, right? That's definitely something you could say. Getting this liver into the test tube is, this one does not want to go in. There we go. Kind of looks like brains a little bit, actually. So we're going to get our livers heated up to their correct temperatures here. And at the same time, we're going to actually do our control of just putting our hydrogen peroxide in a test tube to see what it does when there's no liver. Okay. And by the way that we're going to measure our oxygen is actually just going to be the height of the bubbles, right? Which is not the most foolproof method, right? I'm sure you guys could come up with a lot better ways, but that's going to be the easiest with what we have. And it's going to tell us a lot, right? If the bubble height is incredibly high, that means there's more bubbles. So it will give us that relationship. So find a ruler so that we could do that. Huh. Where are all the rulers? Ooh. All right, sorry guys, there's a lot of materials in this lab, so as you can tell, it's quite a bit to set up here. Okay, so I am monitoring our temps here, especially our body temp liver. We don't want it to get too hot, right? We want our boiling one to actually end up boiling. I'm gonna adjust this so you guys can see the actual stuff here. So our body temp one is getting, it's pretty much at body temp. However, the liver is probably not there yet. right? So I'm going to give it just a couple more minutes. Our boiling one, you can already see the water starting to get some gas inside of it, right? It's getting pretty warm here. We're over 50 degrees Celsius for our boiling. And let's see about our room temperature guy here. All right, so what we're gonna do is take some hydrogen peroxide, brand new, which is good because these can actually expire if you guys weren't aware of that. They do decompose over time. And we're just gonna pour it into an empty test tube and make some observations here about what is going on. All right, did we see any bubbling? No, right? It looks like water, even though it's not. So what we can do is for our data table here, for control, the amount of O2 released, we're gonna write none, right? Our control literally just looks like water. The hydrogen peroxide has nothing to react with, so it's not doing anything. Right now, you can imagine if you pour this on a cut, right? You've seen the bubbling, you've seen and felt the fun times of the bubbling. But in this case, nothing is happening. Okay. Let's see. The other thing you're going to need to be doing is sketching what the lab setup looks like, right? You could do that on paper or you could do that digitally. Really what you're trying to focus on here is that we have these 4 different beakers, right? One that at all of our different temperatures, and that we have the liver inside test tubes inside of a beaker full of water. And that's how we're getting it to the temperatures. So this looks good, actually. Our room temperature water, which we will write our actual temperatures here just as a reference. Our room temperature water is 22 degrees Celsius. Our body temp, actually our freezing, is 2 degrees Celsius. Our body temperature one is probably pretty close to getting done here. Yeah, it's around 40 something. So we'll take it off the heat, let it cool down a little bit. But the liver should be getting very close to body temperature. And our boiling water, we have not actually reached boiling yet. So we'll let that one keep going. So what Jordan and I are gonna do is we're gonna pour the same amount of hydrogen peroxide into all of these test tubes. Why are we gonna have to pour the same amount? Jordan mentioned it. Let's see if someone could tell us in the chat. Why would we want to pour the same amount of hydrogen peroxide every time? Thank you, Bella. It will affect the bubbles, right? Because if you did it, it wouldn't get an accurate result. Exactly, right? If I pour more hydrogen peroxide on one of them, that might be why there's more bubbling, right? So we want to keep everything the same except the temperature. I'm using all glass beakers, right? I'm using 2 thermometers just because one is more for the cold ones and one for the hot ones because that's a little easier. We used the same size liver, right? Ideally, we would want this liver to be from the same chicken, right? It might not be, it probably isn't, but that's fine. All right, so we're gonna do 5 milliliters of hydrogen peroxide. And we're actually going to start with our ice cold liver, and we'll just confirm. It feels nice and cold, right, Jordan? Okay. And I need Jordan to actually measure for me the height of the bubbles. We're going to measure in centimeters, right? So it might just go up to the top of the tube. If it keeps going up, sometimes they actually climb out of the tube. Okay. So let's move this boiling water away from my computer. Okay. All right, so you guys ready? This is the fun part where we actually see bubbling, maybe. So who thinks that the freezing cold, I'm actually warming it, so I'll put it back in there. Who thinks that the freezing cold liver is going to produce bubbles? Does anyone think that? Or we're all going, maybe. Well, let's find out. Okay, ice cold was around 3 degrees Celsius. Let's see if I can actually get my computer closer. I want to have a good video of this.
So if it's cold, wouldn't it make it like more dense? So it'd be harder for the liquid to pierce it and really get into it.
That's a good point, right? If it's colder, maybe the liver has actually shrunken in. All right, let's see. Okay, we got some bubbling. It's like climbing up my finger. I don't know if you guys can see this, but the bubbles are actually coming out of the container here. What do you want to say, maybe 14?
Yeah, 14 and a half.
Okay. So our ice cold liver, actually surprisingly performing pretty well for ice cold liver. 14 centimeters of bubbles. We will now do our room temperature. And Jordan, would you like to fill this up to 5 milliliters, please? Our boiling liver is getting close over here. Now we're in the 80s on our Celsius for our boiling, getting very close. All right, our ice cold liver. Surprisingly, I'm very surprised by that. All right, room temperature. You guys think it's going to be more or less than the freezing cold? Go ahead and type in the chat more or less. Destiny says less. Colton also says less. We got some mores, lesses. Okay. This will be interesting to find out. Now keep in mind too, right? This might not be at exactly room temperature, but it's pretty close.
That's gonna go all over your computer.
Yeah. Move it away from the computer. Okay, so this time we actually have bubbles falling down the side of the container. So would we say that's more? Definitely, right? If we count these peaks of bubbles, I would say it would have come up to here.
Yeah, so probably over. So about maybe 18.
18. I would agree. Okay, so our room temperature liver produced more bubbles than our freezing cold liver and went up to about 18 centimeters. Of course, we are estimating this, right, because we can't actually have a tall enough test tube. For some reason, we only have these baby test tubes here. Let me grab a paper towel real quick. I've got a liver hydrogen peroxide all over me. Fun times. Okay. So up next, we will do our body temperature liver while we're waiting. And I know Jordan had said as part of his prediction, that he thinks body temperature is probably going to be the best, right? That maybe even could have been the control that we compare everything else to. So let's get a final reading here on our temperature for our body temperature. It's a little warmer than I would like. It's about 44 degrees Celsius, which is a tiny bit warmer than body temperature, but I doubt the liver is actually that warm. Okay. So, body temperature liver, nice and beautiful there. Okay, you ready? We got some bubbling, not quite as much. It seems to be going a little slower, right? I might have gotten it too hot. That's probably we could actually try to do it with another piece just to make sure.
Gradual boiling will do.
Yeah. So what, you think it's about 9? 10?
Yeah.
10. So we'll say 10, and in fact, we will use another test tube here. We'll use our control test tube. And we'll try the body temp one more time, just because I think that I did get it a little bit too warm. It's very hard to heat something up to exactly body temperature. You know, the best way to do it is to actually hold something next to your body. But I don't really want to hold liver next to my body, so we will do this other way of putting it in water. Okay, so we'll let it chill in there for a sec and see if that helps a little bit. Okay, our boiling liver, it's going to town over here. It is boiling. Why can't I get it in the frame? There we go. It's boiling pretty intensely. So it's going to be really hot. Try not to burn myself here. Of course, I don't have proper dish stuff in here. This might not work. Okay, let me see. I'll use my hat. Actually, someone left a sweater. Leave your clothes in the science room and I will use them to grab hot items. All right, so check out the liver that was in the boiling water, right? It actually looks slightly cooked, right? Which makes sense because it's been in boiling water, right? And that's okay. That is actually what is supposed to happen. So I'm actually just going to set it in this test tube rack because it's very hot, right? I don't want to burn myself here. And we will see what happens to, can you guys see this okay? Yeah, you can. We'll see what happens to the cooked boiled liver. Do we see any bubbling?
No.
Barely, right? I see like, let's see if I can touch it. I see like a tiny, tiny layer of bubbles. It's very, very, very small.
So if it's too cold, it doesn't do that much. But if it's too hot, it doesn't do any. So that means there's like a sweet spot where it does like a punch.
Exactly, right? There's actually a bell curve for enzymes, right? So if we graphed this, we would see that closer to body temperature, right, we're going to see the most bubbles. And then as we go to the extremes, we're going to see very little to none. If we had actually gotten this liver below freezing, for example, we probably would have absolutely no bubbles. But our cooked liver, right, also barely has any. In fact, I'm going to say that's about, what, 0.1 centimeters? It's like a really tiny amount of bubbles. Almost none. So 0.1 centimeters for the boiling. So we're going to try that body temp one more time. And then we will actually start talking about the conclusions behind this, and then we're going to prove that oxygen is here. Or actually, I thought we were. I might actually have to find a lighter to do it, though. We're going to use fire to do it. Okay. Oh, good. This liver is a little bit closer to body temperature at 39 degrees. I'm going to let it sit in there for just 1 second. How boiling was boiling? Oh, great question. That's actually done boiling now, but it was full on boiling, so we're going to say it was at 100 degrees. Okay, let me see it really quickly. Well, dang, I might not be able to do the proving of the oxygen because I need a lighter. I'm trying to think about where one is. All right, so we might not be able to prove this right now. I might just have to explain how we would do it. And I might actually find a lighter in a few minutes and be able to prove it. But basically, the way that we could prove this is we could have an ember on a piece of wood, right? And if we place it above the reaction, it will actually ignite because of the oxygen, right? Because oxygen fuels fire. So if oxygen is being released, if we place an ember above the reaction, we will see like a little poof explosion. And possibly something, the wood could catch on fire again, which is pretty cool. And I would like to do, so we might actually go on a little trip to look for a lighter. But let's try the room temperature one more time, or excuse me, body temperature, just because I feel that I might have cooked it a little too much. And now that you guys saw what happens with the cooked liver, right, I'm sure that you're convinced too that we should probably check this one again. Okay, so we're much closer to body temperature now. Let's see what happens. Okay, it's like going insane and dripping out of the tube, which is what's supposed to happen. It means I cooked the other one too much. Okay, so we're gonna change our body temp reading. That was more even than room temperature because it actually dripped onto the table. So we're gonna change our body temperature to, we'll say 25 centimeters approximately. Okay, so let me go ask in the front, see if, I think we have one of those grill lighters here somewhere, which would work. I knew I was forgetting something this morning. I remembered my rainwater for earth science, but not the lighter.
-10. And now I've got -10 times -6.5. And every time you multiply it, that's
I'm in a live lesson. I realized that I need a lighter. Do we have one of those like grill lighter ones lighter? Or any kind of lighter?
I feel like there's a lighter in my drawer right here. I'm not sure why.
That would be super convenient.
But it's definitely not a barbecue lighter.
That's fine. It doesn't matter what kind. I just need to light something on fire.
what exactly?
I have 9 kids patiently waiting to see something lit on fire.
Is it just anything?
Yay! Thank you, Hillary. Say hello. You saved the biology lesson. Well, let's not try to pretend I have primitive skills. Okay. Yeah, exactly. All right, guys. So we got the lighter. That means we're going to be able to prove this right now. So we're going to use some room temperature liver because we can tell from our data that that's going to produce some oxygen, right? And we're going to try to get this ember. And it's pretty hard, so I'm probably going to have to do it a few times. because as you can imagine, you got to get it timed completely right when the oxygen's coming up. So I have this, I don't know if I feel safe putting fire near plastic. Okay. Use this guy here. So we'll go ahead and get a nice, large piece of liver. And we're going to pour quite a bit of hydrogen peroxide right at this point. We're not concerned with the bubbles, per se. We're more concerned with proving that the bubbles are oxygen, right? So the amount that we're using here isn't going to matter. I might have to do it multiple times. Okay, so we're going to take our wooden stick, and we're going to get an ember going while the reaction is going, which is where this gets complex. And in fact, I'm going to stop sharing the screen so you guys can see this in big motion picture. Okay, liver is going. We see bubbles. Okay. We have an ember going. Look at that. Caught on fire. Another ember. Oh, I'm so good at it this year. Yay. Caught on fire again, right? That oxygen is fueling that ember to re-catch on fire. Can you guys see that okay at home? I hope so. So that is how we can actually prove, right? And we could just keep doing that over and over, because the interesting thing about enzymes, right, is that they can get used over and over and over, assuming that you don't kill them by boiling them. So these enzymes are going to keep breaking all that hydrogen peroxide down until there's absolutely none left, right? So they'll keep going and going and going and going. And now it smells like nice burned wood in here. And then we could actually use this liver again to do the same thing, right? Because think about it, the enzymes in your body are getting recycled over and over and over. You're not making new enzymes every time you need to do a reaction. That would be insane, right? So the most important takeaways from this are, one, temperature affects enzymes, right? Clearly. When it's really hot, it was boiled, did not work, right? When it was cold, it still worked. We probably didn't get it cold enough. But as you can imagine, if it gets super cold, not working. The same thing is going to be true with pH, right? The pH of your body is 7.4. So enzymes are going to struggle to work at pH is higher than 8 and pH is lower than 7. It's a much narrower range for pH. And salts, right? Salt can depend on the enzyme, but there are certain salt levels as well that we need. So the rest of this lab is actually you just kind of describing your observations, right? You guys might not be able to hear it, but when the reaction's happening, it's really loud, right? There's a ton, it's like, it sounds almost like
you're frying something.
Yeah, like you're frying something, or I was gonna say that cereal.
Oh yeah.
The Snap, Crackle, Pop cereal. What is that cereal?
Rice Krispies.
Rice Krispies. It's like making little popping sounds, right?
Or when you're cooking bacon.
Yeah, or like cooking bacon. What reaction lasted the longest? That's up to you. I would say that the body temperature reaction lasted the longest because it actually bubbled out of the container and kept going, right? And then how did we test for the presence of O2? Well, we just tested by the embers, right? Sticking the ember close to the reaction and seeing it ignite on fire. So the last part here for you guys to do on your own, right, and also possibly finish your drawing and or your vocab, is to answer these conclusion questions. Was your hypothesis supported, right? Were you right about what you said? Explain the data, right? Use the data when you talk about a lab. That's why we did the data, because people want to hear what you got. And then how does this relate to real life? And then explain just how temperature affects the enzyme reaction rate, right? I know some people actually did the explaining after they did the graph. For the graph, you can use a website to graph it, that's fine. You can graph it by hand, you could graph it on the computer, right? Whatever way is the easiest for you to graph. I want you to look at the graph. I don't really care about the graphing process, right?
What does this do?
So this is gonna be due on Monday. Okay, so that gives you today and Monday to work on this. There is a code word for today, which I will tell you, Jordan. Oh wait, I don't want to show people the code word. I did that last time too. I was like showing the code word. Okay. Oh yeah. So the code word is, if you have the code word, you can get up to 100% on the assignment, right? If you don't have a code word, then, oh, I know, I didn't say it. I just put in the chat. People can't see the chat. If you don't have the code word, then you can get a 90%, right? You see a running theme in my code words, yes. Only for this class. My other classes have weird ones. So if, obviously you're not gonna get 100% though if you don't do the whole lab, right? So make sure you answer all the questions to the best of your ability, right? I'll give you feedback if you say something that's wrong. And hopefully this kind of gave you a greater understanding of what is going on internally inside of you, right? Your liver does this all the time. It's probably doing it right now, breaking down hydrogen peroxide for you. Okay. Do we have any questions? Oh, also a very important announcement. Next Friday, we will not have a virtual session, right? I'm also going to send an email about that today. But we will not have a virtual session next Friday. I'm going to be in the East Coast, and the time zone switching is going to be different, and I'm going to be going to a wedding. So it's not going to be very easy for me to do the virtual session. I am going to put a little check-in reflection assignment for you to do that should take you about 10 minutes, just getting you to reflect about how you're doing so far in the class, right? The collaboration project is starting now, right? A lot of people are posting about finding partners, which is great. Keep in mind, you're going to be done with the content of the course in December. And you will have a lot of time in January to redo stuff and also to work on your project, right? So the content, we're like over halfway through the content right now, which is exciting.
So the stuff that we need to know in order to do the project, we don't know all of it yet.
Exactly. You might not know everything yet for the project.
That completely explains why some of them, I was like, wait, what?
Yeah, so the ecosystem option, right? We haven't learned about ecosystems yet. You might already kind of know some things about them. That one is obviously going to be hard because we haven't learned that stuff yet. And also the biofuel one, we're going to learn a little bit about biotechnology, but we're not going to learn too much about biofuel. That one will require a lot of your own researching, right? But if you like researching and that's something that interests you, then that's great. Yeah. Options 1 and 2, we've already learned that content, right? Osmosis and diffusion, we already talked about. And also cellular respiration and photosynthesis, we already talked about. So if you want to start your project today, those would probably be the better ones to choose. But the ecosystem stuff is pretty easy in terms of content, right? It's not molecular, it's big picture, and it's a lot of vocab like producer, consumer, decomposer, stuff that you've probably already heard or that you're going to be like, oh yeah, I know that. All right, so any other questions? This is the write-up. Yes, Ariella. So you're going to turn in this document with your questions and stuff answered on it. And the data is going to be in the document that's posted, right? Oops. So in the document that's posted, which Oh yeah, you guys couldn't see that. I forgot I wasn't sharing my screen. I was like, oh no. In the document that's posted, there will be the data in case you didn't get it written down or you need to double check it. So really, you're responsible for the sketch, the conclusion questions, and the pre-lab questions. Okay, well, thank you guys for coming and watching some liver enzymes in action here. And I won't see you guys next Friday, but I'll see you in 2 weeks. Have a great weekend.